Joo Cheon. I'm an associate professor in Albany Medical College who's working on ovarian cancer research. So today I'm going to, this one, this title is my kind of official, like official research title. When I have to give formal presentation to other institution, then basically this fancy title is what we what we what our research is about. But today I'm going to talk about like three parts a bit. So first part I'm going to talk about what ovarian research we are doing, and then second part I'm going to kind of briefly kind of introduce you how we perform cancer research in the lab, and then lastly it's more kind of personal. It's a little bit kind of personal story. So for those who are interested in becoming cancer researcher, I'm going to kind of briefly explain why and how I became a cancer researcher. So, so let me talk about my research first. So, what is cancer? So, do you guys know what cancer is? Are you familiar with what? Are you familiar with cancer, or do you know anyone who has cancer?
Yeah, I think I know briefly what cancer is, and I've met a few people who've had it.
Mm-hmm. Yeah.
Yeah.
So nowadays, like so many people like have cancer. So yeah, so basically like cancer, as you guys might know, cancer is the disease in which some of our body cell uh, become abnormal and then grow nonstop, grow uncontrollably. So usually our normal body cells, if they are if they grow uh, if like they grow too much, then they stop growing, and then even some of them undergo cell death, so that like yeah their growth can be controlled. But once normal cells turn to tumor cells, then even if these tumor cells like they just continue to grow and grow, and then yeah nothing can stop cancer cells growth. So this cancer cell kind of growing or continue to grow and grow, and then if the space is limited, then they even start to invade and spread to other distant organs, and then grow there. So once they grow, then they grow bigger and bigger, and then if there's not enough space, then they start to invade and then make colony out in other organs, then start to grow in distant organ, and then again the grow there, and then eventually this unlimited growth of tumor cell eventually kind of block normal function of organs. So that's why like people die. So cancer, and then also cancer can start almost anywhere in human body. And then cancer is also very common. So cancer is the second leading um, cancer is the second leading cause of death in United States. And do you guys know what is the leading cause of death in America? What disease is the leading cause of death in America? Do you guys know?
No, I don't.
I'm not sure, but can I guess? I feel like it's diabetes.
Similar heart disease. So heart disease is the number one killer, and then cancer is the second leading cause of basically cancer is the second killer, and every year about 2 million people 2 million people are diagnosed with cancers, and about 600,000 people die of cancer. And then, as you can see in the below graph, lung cancer is the number one killer. And then, like for example, for ovarian cancer is the fifth leading cause of cancer death among women in United States. So basically, cancer is quite common and then kill lots of people. And then the cancer I study is ovarian cancer. Basically, ovarian cancer is cancer that begins in the female reproductive organ that produce egg called ovary. So yeah, so you can see ovary. So basically, a woman has two ovary, and then again, ovarian cancer is the cancer that arise from the ovary. That's the ovarian cancer. And then, as I said, ovarian cancer is the fifth leading cause of cancer death among women in United States. And then, uh, ovarian cancer is the most deadly gynecological cancer. And then in America, about 20,000 women are diagnosed with ovarian cancer, and 13,000 women die of ovarian cancer every year. And then if you are a woman, then 1 in 78 women has a risk of developing ovarian cancer in their lifetime. And then another striking thing about ovarian cancer is that despite all the advance in surgical technique and drug development, survival rate have not been substantially improved over 40 years. So 40 years ago, like 5-year survival rate was 35%, but 40 years later, the survival rate is like 49%. The kind of minor improvement but it's not kind of substantial improvement. And then this is how kind of ovarian, and then this is basically showing like a different stage of ovarian cancer. So for example, stage 1 is basically like when tumor cells just like limited to the ovary, then that will be stage 1. But when like tumor start to spread neighboring female reproductive organ and remain in pelvic cavity, that will be stage 2. And then when tumor spread to abdominal organs, like up to diaphragm, then that will be stage 3. But if tumor spread to beyond the lung cavity, then that will be stage 4. And then like initially stage 1, the survival rate of stage 1 is over 90%. But unfortunately, majority of women are diagnosed in advanced stage, such as stage 3 and stage 4, which survival rate is less than 30%. And then, as I said, ovarian cancer is like most lethal gynecological cancer. And then you might ask why ovarian cancer is so lethal. Then there are two main challenges. So first one is like early detection of ovarian cancer is very difficult. So as I said, majority of women are diagnosed in stage 3 and stage 4. The reason is that, like, number one, symptoms are quite vague. So as you can see, the ovarian cancer symptom is like bloating, urinary frequency, pelvic or abdominal pain, difficulty eating or feeling full quickly. So as you can see, these symptoms are quite vague and general. So this type of symptoms are often ignored. And then the biggest problem is that we don't have any good early detection marker nor annual screening test. That's why ovarian cancer is diagnosed so late. And then also pap smear does not detect ovarian cancer. And then although we have like a blood test called, even though we have CA125 blood test to diagnose ovarian cancer, usually CA125 blood test has like lots of high false positive rate. Therefore, this cannot be used to detect ovarian cancer early, and then it cannot be used as a screening test. And then the second problem of ovarian cancer is that ovarian cancer develop chemotherapy resistance quite quickly. So when women are diagnosed with ovarian cancer, usually they receive debulking surgery followed by standard chemotherapy. But unfortunately, 1 in 5 patients, they do not respond to standard chemotherapy upfront because of their intrinsic resistance to standard chemotherapy. Therefore, they do not respond to this standard chemotherapy and die shortly. And then for the remaining patient, the standard chemotherapy work well, and then sometimes even tumor disappear. But unfortunately, within 3 years, more than 75% of patients, like tumors come back, And then this time, this tumor acquire resistance to standard chemotherapy. And then once this patient develop resistance to standard chemotherapy, then we have very limited treatment option for them. So that's why like ovarian cancer is so lethal. So so basically research interest in my lab is basically to understand the molecular mechanisms by which ovarian cancer cell become more metastatic and more chemotherapy resistance, um, especially through the interaction with the various components of the tumor microenvironments. So actually, if you look at the tumor cells do not exist alone. So as you can see here, tumor cells, if you look at tumor cells on tumor under the microscope, actually tumor cells are surrounded by so many different neighbors. For example, tumor cells are in tumor microenvironment. So that means like that's the environment where tumor cells live. There are so many cells and components. For example, there are blood vessels, so that cancer cell can acquire oxygen and nutrient from this blood vessel, and then various inflammatory cells, and then fibroblast cell, and then like there are some kind of extracellular matrix proteins. Yeah. So basically, this cancer cell constantly interact with these various components of their environment, and then they kind of influence each other's, and then so that cancer cell affect tumor microenvironment, and tumor microenvironment can also affect the tumor cell phenotype. So they are constantly crosstalk each other, so that to make more friendly environment for cancer cell. So basically, my research focus on like how ovarian cancer cell communicate with the environment, and then if we understand that molecular basis, then we can block that communications to kind of to make cancer cell more sensitive to standard chemotherapy. So ultimately, our goal is once we understand the molecular basis, how ovarian cancer cells communicate with the environment, then we specifically block those communication to make cancer cell more sensitive to standard chemotherapy. And then eventually we want to develop novel personalized biomarker-guided targeted therapy to improve survival outcome of ovarian cancer patients. So toward this goal, at this like right now, yeah, we have like 3 research projects ongoing. But for today, I'm going to talk about only like first project, which is centered around like one novel biomarker we identified, the COL11A1. So as I said, like like initially, like ovarian cancer patients were sensitive to standard chemotherapy, but eventually they will develop chemotherapy resistant. And then, like our goal was like, how can we identify this poor outcome patient who will develop chemotherapy resistance as soon as possible, so that if we can identify this type of patient as early as possible, then maybe we can enroll them into clinical trial, or if we understand what biological pathways are upregulated in this patient, then we can block those patients, we can block those pathways as a kind of like targeted therapy to improve survival outcome of this patient. Therefore, we were kind of asking what biomarker genes can we use to identify this poor outcome patient, and then what biological pathways are upregulated in these patients. So to answer the questions, we analyzed molecular profiles of 710 ovarian cancer patients. And then basically, we divided into 2 patient groups, patient with good survival outcome, and then patient who have poor survival outcome. And then we were comparing molecular profile between 2 patient groups and see which genes are upregulated in this poor outcome patient. And then basically, we identified 10 genes are commonly upregulated, 10 proteins are basically upregulated in this poor outcome patient. And then this is the list of those 10 genes commonly upregulated in these poor outcome patients. And then surprisingly, they are all of them are involved in the same biological process that basically all these tangents are basically are about kind of depositing collagen and then weave collagen fibers to make stiff collagen matrix around the cancer cell. So all these tangents basically contribute to that biological process, and then somehow that process contribute to poor survival outcome in ovarian cancer. And then you might ask, what is collagen? Then actually collagen is most abundant protein in our body, and then most collagen is commonly found in bone, muscle, skin, and tendon. And then our skin is like very stretchy because of collagen. And then, but like, yeah, collagen is very important in our body. But the thing is, like in cancer, like cancer cell also like in normal situation, of course, collagen is also very important to provide strength and structure of our body. But during cancer progression, cancer cell like upregulate various like abdominal collagen fibers to make collagen matrix more stiffer and more linear around the cancer cell compared to normal tissues, and then that kind of somehow makes more friendly environment for cancer cell. And then basically the tangents we found is basically like upregulating some abnormal collagen subtype and then making then and then enhancing enhancing kind of like weaving collagen fibers each other so that it makes more kind of stiff collagen matrix. And then actually like we also found that upregulation of those tangents are also associated with not only in ovarian cancer but other solid cancer as well. So high levels of tangents are associated with like poor survival outcome and more aggressive tumor phenotype in multiple cancer types such as lung cancer, head and neck cancer, pancreatic cancer, etc., suggesting that this type of like making like depositing more collagen and then making more like a stiff collagen matrix around the cancer cell that contribute to poor survival outcome and more aggressive tumor cell phenotype in multiple cancer type. So it's more kind of general phenomena. And then this is kind of like consistent with clinical observation that clinical observation. So for example, women take mammogram. So basically, this picture shows like a mammogram of the breast, and then actually like the collagen fibers can be like visualized, and then actually like this type of like dense collagen fibers, like you can see the wider area. That means like more dense collagen fibers, and then this one is a kind of high risk factor of breast cancer, and then indeed like this type of like a dense, stiff collagen fibers and matrix that kind of promote like breast cancer progression. And then actually like our discovery of tangent like is consistent with this type of like observation in breast cancer. And then among those 10 genes, we became particularly interested in one biomarker gene called collagen 11A1 because collagen 11A1 is the most significantly upregulated gene during ovarian cancer progression. And then basically these are tumor tissues obtained from the same patient at different stage. So primary means tumor just stay in the ovary. Metastasis means that tumor spread to other organ, and then recurrent tumor means that after like she got treated with standard chemotherapy, and then when tumor came back, then that means that like that's the recurrent tumor. And then this brown dot indicates collagen 11A1 mRNA signal. Then as you can see, in primary cancer, not much brown signal, but in metastatic ovarian cancer, you start to see a little bit of brown, But in recurrent ovarian cancer, when ovarian cancer come back, you see highest levels of collagen 11A1 signal. So basically, you can see significant upregulation of collagen 11A1 during ovarian cancer progression, kind of suggesting that collagen 11A1 might be important for ovarian cancer progression. And then you might ask, what is collagen 11A1? So actually, like collagen 11A1 is a kind of like one of the three alpha chain one of the three alpha chain of collagen type 11, which is a minor fibrillar collagen mainly expressed in cartilage. And then actually collagen 11A1, most studies on collagen 11A1 was done in bone development, because if there are mutation in collagen 11A1, then like for example, if mouse don't, if there's a mutation in collagen 11A1 gene, then mouse die at birth because of severe skeletal defect, suggesting that collagen 11A1 is very important for bone development. And then actually, like if you see, like if you look at those mice more carefully, and then what people found is that the mouse who have like a mutation in collagen 11A1 gene, they have extremely thick collagen fibers and fragmented collagen fiber, suggesting that collagen 11A1 is also important for proper collagen fiber assembly and organization. But yeah, but then this collagen 11A1 is important for bone development and proper collagen fiber assembly, but somehow this collagen 11A1 is suddenly hugely upregulated in cancer cell, and then that contribute to like more aggressive cancer cell phenotype. And then also, like as you can see, basically if we look at tumor, ovarian tumor under the microscope, and then actually like when there's the collagen 11A1 positive area is associated with like in collagen 11 positive area, you can see thin linear collagen fiber, whereas collagen 11 negative area is like you can see this type of thick wavy collagen fiber, again suggesting that collagen 11A1 might kind of contribute to this making this type of thin linear collagen fiber. And then it has been shown that this type of thin linear collagen can serve as a like highway for cancer cell to migrate and spread to other organs. So probably that's, yeah, this is one of the reasons why collagen 11A1 might kind of promote more aggressive cancer cell phenotype. And then like in terms of like collagen 11A expression in tissue, we also found that cancer cell itself express like pretty low amount of collagen 11A1. Actually, collagen 11A1 expression is more abundant in the just adjacent environment. So especially like a fibroblast cell adjacent to tumor cell, they really express really high levels of collagen 11A1. That means that tumor cell and fibroblast cell adjacent to tumor cell, like they kind of like communicate each other to upregulate collagen 11A1 in the environment. And then we also found that like the chemotherapy resistant ovarian cancer cell also upregulate really high levels of collagen 11A1 too. And then like, so to understand like what collagen 11A1 is really doing in ovarian cancer cell, what we did was like we depleted collagen 11A1 from cancer cell and then checked cancer cell phenotype. And then we saw that if we deplete collagen 11A1 from cancer cell, then cancer cells show significant decrease in cancer cell migration, invasion, and tumor spread to other organs. So in other words, without collagen 11A1, cancer cell cannot migrate, cannot invade, cannot spread to other organs, suggesting that collagen 11A1 is not just biomarker, but also a very important player in ovarian cancer progression. And then actually like from patient data, we also learned that collagen 11A1 is one of the top upregulated gene in chemotherapy resistant patient compared to chemotherapy sensitive patients. So therefore, we also checked whether collagen 11A1 actually directly contribute to chemotherapy resistance, and then the answer is yes. Yeah, collagen 11A1 also make, if cancer cells are cultured, grow on the collagen 11A1, then these cancer cells become more resistant to standard chemotherapy. Then we were asking, okay, then what is the molecular basis? What's the mechanism by which collagen 11A1 make cancer cell more resistant to standard chemotherapy? And then our analysis basically found that, so again, the cancer cell kind of communicate with the fibroblasts, and then these fibroblasts, in response to signal from cancer cell, they upregulate collagen 11A1 and then put collagen 11A1 in the environment, and then and then cancer cell binds to this collagen 11A1 through cell surface receptor, and then activate like pro-survivor pathway, and then one of them is pro-survivor pathway, which is kind of block chemotherapy to block programmed cell death induced by chemotherapy. So that's why, like with collagen 11A1, cancer cell can survive better despite chemotherapy treatment. And then another mechanism we found is that when cancer cells binds to collagen 11A1 matrix, then they activate pro-survivor signaling and then also upregulate fatty acid metabolism. So usually, like cancer cell use sugar, glucose, as their main energy source. But somehow, when cancer cells grow on collagen 11A1, they switch it from glucose to fatty acid as their main energy source, and then they breaks down fatty acid to produce energy or biomolecule, and then that also contribute to chemotherapy resistance in cancer cell. And then if we block fatty acid metabolism, then cancer cells become more sensitive to chemotherapy, even though they are growing on collagen 11A1. So then, like, then we thought, okay, that's great. Now we can use collagen 11A1 as a biomarker to identify patient group who will not respond to standard chemotherapy. Then we can kind of block fatty acid metabolism pathway to treat them. So that's what we did. But actually, like, we realized that just blocking fatty acid metabolism alone is not super effective in killing cancer cell. And then we were puzzled at what's going on. And then further study, we kind of found actually cancer cells are quite smart. So when cancer cells grow on collagen 11A1, actually they activate two parallel signaling pathway. So one signaling pathway is like upregulating fatty acid metabolism, but the other signaling arm is like activating Hisha protein 27, which is a stress-induced protein. And then what happens is, like, if we block fatty acid metabolism, then cancer cells upregulate Hisha protein 27 to compensate for the loss of fatty acid metabolism. Vice versa, if we block Hisha protein 27, then cancer cells upregulate fatty acid metabolism to compensate for the loss of Hisha protein 27, and then they can sustain survival. But if we block both pathway, then like we can actually induce significant cell death of cisplatin chemotherapy resistant cancer cell. So for example, we can block fatty acid metabolism using the drug called paraxylene. So that's the drug being used to treat like angina, which is a heart condition. And then we can block Hisha protein 27 using the drug called like ivermectin. So as you can see, if we just use the single drug blocking one pathway, it's only 50% effective in killing chemotherapy-resistant cancer cell. But if we combine both, just blocking both pathway, then we can almost kill entire cancer cells. So this combination therapy seems to be quite effective in killing chemotherapy resistant cancer cell. Therefore, right now we are kind of excited. So that means we can identify patients using collagen 11A1 as a biomarker, and then we can probably treat those patients using this type of combination therapy. So again, to summarize my research, basically, probably we can use collagen 11A1 to identify patient group who might have poor survival outcome, and who might develop chemotherapy resistance later. Then once we find this type of poor outcome patient as soon as possible, then probably we can treat them with like fatty acid metabolism inhibitors or this type of like ivermectin paraxylene combo, so to make cancer cell more sensitive to standard chemotherapy. So that's kind of our ongoing research. So that's part one. So up to this part, do you guys have any questions? Okay, then let me move to the second part. Then how do we perform?
Go
Yeah.
ahead, Julia. Do you have a question?
Yeah, sorry. I wasn't sure if you mentioned this before, but is there an easy way to test for the collagen levels?
Oh, that's a good question. I think yeah. So what we actually, like, the best way is, so when ovarian cancer patients are diagnosed with ovarian cancer, then usually like they have to get the surgery, because so, and then during the surgery, if we can get those kind of like excess tumor tissues, then we can grind them up and then kind of measure collagen 11A1 mRNA levels, or we can fix those tumors and then stain them with collagen 11A1 protein. So in this way, yeah, we can find who will express higher levels of collagen 11A1 among other patients, and then we can probably treat them with this type of targeted therapy. So the answer is like, yeah, once from the surgery, we can get some kind of tumor tissues, then we can measure either collagen 11A1 mRNA levels, or like using real-time PCR, or we can make paraffin section of those tumor tissue, and then stain them with collagen 11A1 protein. In this way, we can measure collagen 11A1 level in patients.
Cool, thank you.
Okay, then let me just move to the part two. So far I talked about like what ovarian cancer research do I perform. The second part is like, how do we perform cancer research in the lab in real life? So this is basically the photo showing us in the lab how we perform cancer research in the lab. So you can see like me looking at microscope. You're looking at microscope to see like a patient tumor under the microscope, and then also like we can label tumor cell with like green fluorescent protein, so that basically we can make tumor cell glow, and then like we can observe those kind of glowing tumor cell under a special microscope, or like and then we kind of we can grow cancer cell in this kind of this plate, and then once we grow cancer cell in the plate, then we can add like genetic material, we can treat them drugs to test cancer cell response to those kind of like genetic material or those various drugs. And then lastly, my technician is doing some kind of like a protein analysis. So this is kind of like just basically photo showing you how we perform cancer research in the lab. So to speak more specifically, as I said, first we commonly use like we kind of commonly grow human ovarian cancer cell in this type of plate, and then we can easily expand them, and then we grow this human ovarian cancer cell all the time, and then we also do kind of molecular analysis, and then after we grow them, and then we treat them with various drugs, and then we harvest the cancer cell and then do various DNA, RNA, protein analysis. And then also we can stain tumor cell with like a various protein, and then each protein can be stained with like a specific antibody conjugated to a fluorescent marker. That's why basically, you can kind of stain tumor with like a various marker. So for in this case, like cancer cells are basically stained with like blue, and then like cell adhesion molecule called E-cadherin is like labeled as green, and then actin is stained as red. So you can kind of do this type of imaging analysis just to see okay, how cancer cells express like a various molecule to support their growth, that kind of stuffs. And then we can also use like fancy microscope to do live imaging of cancer cell real time. And then, for example, this one, like you can actually see like cancer cells kind of like a squish and then move in real time. So, and then you can also see that like this type of cancer cell use bubble-like structure to move forward. So we can use this type of live imaging to like study cancer cell behavior in living organism real time. So this is another way to study cancer. And then also after we grow cancer cell in the plate, then we inject this cancer into mice so that cancer cells grow in the mouse, and then we treat mouse with various drugs and then measure tumor size. In this way, we can kind of monitor like tumor cell growth and tumor cell response to certain drug in living organism. So mouse model is quite important for cancer research. And then also like we obtain patient tumors from operating room, and then either we isolate a human tumor cell from patient tumors, or we fix them and then like stain them and then look at them under the microscope. Or lastly, we can like download some public cancer data. We can download some data from public cancer database, and then we can kind of reanalyze those kind of cancer database to get some kind of to find some kind of interesting information from this kind of public database. So that's how we perform cancer research in the lab. And the last part, I'm going to talk about like why. It's kind of more personal story. So why and how did I become cancer researcher? So this type of information might be interesting to those who are interested in becoming cancer researcher in the future. So why did I choose to study cancer? So well, actually, like when I was a high school student, as you guys, at that time, like actually, like I wanted to go to the medical school because I thought like, saving patient life is so cool. And like, for example, like at that time, I was so fascinated by the drama called ER, and then all these cool doctors like save lives, or save patients' life, and then I thought that was so cool. But so yeah, that's why I wanted to go to medical school. But unfortunately, I took national college entrance exam, and then I didn't get high scores good enough to enter medical college. Therefore, alternatively, I chose nutrition major. But during the first 2 years of college, nutrition major was kind of boring to me, and then I started kind of like puzzled what should I do next? Why I live and what I live for? What I need to do after graduation? That kind of stuff. So I was searching for those answers seriously, and then I found myself. Then I kind of found the answer that, okay, you know what? I want to do something meaningful and helpful to others, so that my life won't be useless and meaningless. Then what should I do, like, to be helpful, to help people as much as possible? Then I decide to do biomedical research to find cure for human disease, so that I can make significant impact on many people's lives. So I thought biomedical research is pretty cool, because if you become a medical doctor, then save you can only treat one patient at a time. But if you can find cure for particular disease, if you can find cure for cancer, then you can save lives of all cancer patients. So I thought, okay, then maybe I should do biomedical research, so that um, I can I can make positive impact on many people's life, that kind of stuff. That's why, like, I chose cancer research. Um, so then, yeah, so that's what I did. So college years, I kind of kept searching, and then finally, okay, I kind of found myself that, okay, I want to do biomedical research. Then what's next? So after I graduated from college, then okay, I decided to go to the graduate school to study. I decided to go to graduate school. So the reason is, yeah, right now my undergraduate major is nutrition, and then in order to do biomedical research, then like I need some kind of transition. So that's what, and then also at that time I was very interested in virus, like Ebola virus, HIV, that kind of stuff. That's why I decided to go to graduate school for my master degree to with a like microbiology major. And then particularly I kind of studied how bacteria such as E. coli or like yeast, how those kind of bacteria or how like E. coli and like yeast can protect themselves from oxidative stress, that kind of stuff. I studied that as a master degree, and then after I got master degree, then I really wanted to pursue biomedical research more seriously. Therefore, I decided to go to America and join the PhD program in United States in MD Cancer Center to study cancer. And then during my PhD study, I worked on ovarian cancer antigen CA125. So basically, CA125 is a blood marker that clinicians use to diagnose ovarian cancer. So if CA125 level is high in the blood, then there's a yeah, that's uh, then that means that you have ovarian cancer. So CA125 is a very useful like cancer marker, but the biological function of CA125 is largely unknown. Therefore, for my PhD study, I generated mouse model that cannot the mouse model who cannot produce CA125, and then see what happened to those mice. That was my PhD study. And then during my PhD study, I was kind of shocked to realize that um, there are so many unknown in ovarian cancer research field. And then also, despite all those kind of effort, still like survival outcome of ovarian cancer patients have not been substantially improved. And then the biologies are so complex. Therefore, after I got my PhD, then I decided to continue ovarian cancer research, hoping that like my research one day can like improve survival outcome of ovarian cancer patients. So this is more kind of my like education and experience, like in formal way. So like I got my undergraduate study in Korea in nutrition major, and then I got my master degree in microbiology in Korea, and then I came to United States, and then I got my PhD in MD Anderson Cancer Center, and then between master and PhD, I also did some kind of like research assistant in Korea Research Institute of Bioscience and Biotechnology to broaden my knowledge and skills in biomedical research. And then after I got PhD, I also did postdoctoral training in LA. And then after that, I came to Albany Medical College to become a professor. So basically, this is like this is Albany Medical College website in showing my research interest, and then as I said, I joined the Albany Medical College in 2015, and then right now, like I'm kind of associate professor working in ovarian cancer research, and then the lab is located in MS Building 338, and then importantly, we are open to any kind of high school interns. So if you guys like are interested in doing some kind of research internship, kind of exposing yourself to cancer research, then feel free to email me. So okay, then for those who want to become a cancer researcher, then what do you need to do to become a cancer researcher? Then the first of all, when you go to college, then you need to take lots of biology class, you need to take biology, molecular biology, cell biology, biochemistry, physiology, genetics, etc. Like lots of biology related courses, and then also I strongly recommend you to do some kind of research internship in the laboratory. So in this way, first of all, like you can actually like you can learn actual kind of like you can get good sense whether research is the right career path for you or not, and then so yeah, and then also like once you have more research experience you have, you have like a better chance to be accepted into graduate school too. So definitely, research, like kind of build like kind of expose yourself to various research internship opportunity to learn more research, to get some kind of more research experiences. And then if you want to go to graduate school, then of course you need to take GRE general and GRE subject in biology. And then you also have to provide personal statement on why you want to study cancer, or like in particularly which area I'm interested in, that kind of stuff. But at the same, but just to be realistic, this is kind of like kind of timeline. So for example, 4 years of undergraduate, and then it will be like 5 or 6 years of PhD degree, and then after PhD, then you have to take on a kind of postdoctoral training, another 5 or 6 years of postdoctoral training. Then after that, either like you apply to college to become a professor, or you can even go, you can go to pharmaceutical company or bio company, or you can work for government, like or patent journals, etc. So there are many career paths, but definitely PhD is kind of required. So you can kind of consider that. And then of course, like cancer research is not easy to do. There are lots of frustration and failure. And then we put lots of effort in performing experiment. Unfortunately, those experiments, there are lots of negative data, and then so we have to keep optimizing those experiments to get the data, like that kind of stuff. And then also cancer research is very competitive field. And then like job market and research funding is getting tough. And then also it's hard to balance with your personal lives. Therefore, it's kind of frustrating, sometimes very frustrating. And then But in the end, you ask yourself, then you realize that, well, nonetheless, despite all this challenge, I still want to continue cancer research because you know that you can save many people's life with your research. And then also people will appreciate your effort in cancer research and then say that, oh, wow, you are doing cancer research. You are so cool. Or your research is so cool, so important, that kind of stuff, that kind of people acknowledge that. And then also many people will also help you out throughout the journey to achieve your goal in finding cure for cancer, that kind of stuff. So it's tough, but at the same time, it's worthy. So my personal advice to you is that don't think too much about like depressing situation. Just do whatever you can do today, one step at a time, one day at a time, and then just keep trying and then expose yourself to very like many different experiences to figure out what you are really passionate about. And then once you figure out what you are passionate about and what you really want to do, then never, never, never give up. And then lastly, um, yeah, I know these days, yeah, it's kind of like very tough these days. Like especially after pandemic, everybody have like hard time. So yeah, just be kind to yourself and to others. You are doing great job, and then just don't give up. Just do whatever you can. Just and then hopefully, yeah, one day, yeah, you can. Some of you might kind of do some kind of cancer research and find cure for cancer, hopefully. So yeah, so best wishes on your own journey, and then hopefully you enjoyed my presentation. And then if you have any question, feel free to email me. And then so thank you so much for your attention. And then now I will be happy to take your questions and comments. Thank you.
I was wondering like how your like research environment is and like how collaborative your research team is.
Uh, well, so basically cancer research, yeah, so by nature it's very collaborative because like yeah, as because like we can do only limited, so one as a one person we can only do so certain just limited thing. We cannot do like every experiment. In that case, like we need to reach out. For example, I want to do certain experiment which we are not familiar with. Then we reach out to someone whose expertise in that technique. Then like oh, like then we kind of collaborate each other so that like yeah, so that we can get some data from those using those technique we are not familiar with, and then we kind of publish paper together, that kind of stuff. So yeah, and then yeah, we especially like for example in Albany Med we have like several professors, several research lab in the department within the department, and then we always kind of like talk to each other, and then we sometimes we borrow some reagent from other lab, and then vice versa, and then we also discuss if like some experiment don't go well, then like we reach out to people and then like get some advice. So it's very kind of collaborative. And then in the lab we have like so 3 full-time members and research interns, and then they always kind of help out each other. Yeah.
Thank you.
Sarah, you have questions? Hello.
I don't know, the microphone wasn't better. I was just wondering, like, because I'm having difficulty myself, like, coming to terms with this, but how, like, I think you did touch on it. How did you, like, specifically, like, even within ovarian cancer, like how did you decide what you wanted to research and everything?
Oh, you mean, so your question is like what research topic do I choose or among?
Yeah. Just like how did you settle like find your passion in researching ovarian cancer like this specifically? I don't know how to explain it.
Oh, okay. Oh, well, so actually like so for example, when I was an undergraduate student, I had a very kind of vague idea that yeah, one day I want to do a cancer research. And then when I joined the PhD program in MD Anderson, then like I did I had like a lab rotation, the rotation of the three laboratory, and then one of the laboratory, basically that lab is basically kind of studying how like sex is determined between men and women using mouse model. But actually, that lab had like a side project on ovarian cancer, and then I thought, oh, maybe I have to join this lab because I want to do that. I want to take that ovarian cancer project. So then I started to work on this ovarian cancer project and then realized that wow, there are so many challenges and there are so many unmet needs in ovarian cancer field. Therefore, I I decided to continue my career in ovarian cancer research field.
Okay, thank you. That actually kind of helps me. Thank you.
Yeah. Kimaya, did I pronounce your name correctly?
Hi, it's actually my sister's email, but I signed in on hers. I was wondering like how exactly do like your interns contribute to the research?
Well, so for example, so usually like, yeah, when like high school student contact me, then usually like we kind of like we just kind of set up a meeting and then I just go over usually like I introduce like our current research project to those kind of students and then usually like so during the internship then usually like those high school students are kind of like trained to learn kind of some specific techniques commonly used in the laboratory to perform cancer research and then also like usually I give the student like a small side project. So small kind of some pilot project so that like the high school student perform some kind of experiment to get some data. Then usually like I give them some kind of like pilot exploratory kind of small project. So if those high school students kind of generate some data, then it kind of help us. Oh, this data is very interesting. Maybe we should put more effort in doing more research on this topic, or opposite. Uh, Based on this data, maybe this protein might not be important in ovarian cancer cell. Therefore, we need to move on to other direction, that kind of stuff. So actually, like you guys are playing important role in helping us decide like which direction or which direction we should go. Did I answer your
so much.
question?
Yes, thank you so much.
Any other questions or comments? Okay. Yeah.
Okay, if no one has any questions, then I think we're all good. Thank you all for joining.
Yep.
And don't-
Again, yeah. If you have any questions, then just please feel free to email me.
Yes, and don't forget to fill out the Google form that's in your email, guys, and I will email you a completion certificate. Thank you all for coming.
Okay. Thank you.